You're at the cutting edge of RNA therapeutics. I'm John Cooke. I'm professor and chair of the Department of Cardiovascular Sciences here and director of our Center for RNA Therapeutics. And joining me in the studio is Dr. Christopher Brannon, who's a faculty member in our Center of RNA Therapeutics, and he's going to introduce our guest.
Thanks, John. So today we're excited to have Dr. Don Johnson from Croda Pharma joining us. Don received his BA in chemistry at Occidental College, then completed a postbachelorate fellowship at the NIH before obtaining his PhD in biochemistry at the University of California, San Diego. Don was a principal scientist at Bristol Myers Squibb, where he led the development of nucleic acid-based solutions and pioneered efforts in high-throughput screening of nucleic acid lipid nanoparticle formulations. So now with 10 years of industrial experience, he's in formulating liposomes and lipid nanoparticles. He's currently the technical business development manager at Croda Pharma, where he works to establish technical relationships and collaborations with industry and academic partners, including some of us here at Houston Methodist. Today he'll be telling us about innovative lipid solutions for drug delivery systems. I guess we'll just start with a little bit of background. If you could provide us with how you got involved in lipid-based drug delivery systems field and where you think the landscape of the field is headed and what excites you in these systems.
Well, thank you for inviting me. Really appreciate the opportunity to chat here. What I will say is what really drove me into this space is my prior background as a cancer survivor in looking for solutions for treating cancer, for treating diseases such as cancer. And that's really what drove me towards, getting my PhD in biochemistry and what have you. I will say that what I, worked on prior to, starting in industry in the liposomal and the LNP space, this work here is definitely exciting. There's a lot of sort of arms and different functions and sort of mechanisms that we can really utilize to go after a lot of these very difficult diseases that we've normally and traditionally tried to solve through small molecule biologics and sort of other routes as a result.
Don, it's so great to have you on the show, and we share your enthusiasm for the field. We really do think that RNA therapeutics are going to change the way we deliver drugs, the way we develop drugs, and is going to have some really transformational effects in many areas of medicine. But to deliver those RNA therapeutics, we need better lipid nanoparticles, and that's what you're going to be talking about today. So really excited to hear about that. Before we get started on your presentation, I want to just let people know that they can call in questions to us. If you're at your computer terminal, just go to pollev.com. That's POLLEV.com and enter DeBakey, and that will allow you to ask Don questions. Alternatively, if you're on your phone, you can just text DeBakey to 37607. That number is 37607. You text DeBakey and then ask us your question, and we'll ask Don whatever you text in to us. Don, you're talking a little bit about the motivation for what got you into the field. This is a field that's really changing rapidly. A lot of new companies entering in because this idea of delivery with lipid nanoparticles is so important for this transformational new field of RNA therapeutics. Tell us a little bit about the industry, about the players in the field. What are your observations there?
So there are definitely a lot of players in the field, especially centered on the coast. If you look in Boston, you can almost throw a rock and you'd hit a company that is a major player in the space right now. One of the companies I look at, especially top three, would be Tessera, Verve, and CRISPR Therapeutics. I would even place Verve at the top of that because of a lot of, the sort of progress they've made with their pipelines and their platforms. And there's a lot of great data that's going to be coming out, hopefully soon, from these companies, from other companies, that are really going to sort of pave the way for how we address RNA therapeutics. Because the way that LNPs, lipid nanoparticles, have been used so far, and they've been successful so far, have been as vaccines.
Yes.
And so, the next phase is being able to develop these RNA therapies, RNA-based therapies that will, be able to target to cells in treat cases like sickle cell, which is one of the indications, for example, that's very exciting to see progress in with some of these companies. And hopefully we'll see a great positive response from the insurance companies too as well.
That's a really great point, and that you're absolutely right. Lipid nanoparticles, RNA therapeutics have worked great for vaccines, for infectious disease vaccines, for neoantigen therapy for cancer. For therapeutics, I do think we need better vehicles that are less inflammatory and less targeting the liver, targeting other organs. So love to hear your thoughts about that in a moment with your presentation. You mentioned the company Verve. Yeah, we are following them with interest here because our RNA therapeutics program has emanated from our cardiovascular sciences department, and so anything that to do with cardiovascular and RNA we're quite interested in. I know they've got some really interesting data using a gene editing approach to knock down PCSK9 as well as ANGPTL3 to lower cholesterol. So those look very promising, those therapies.
Yes, and what we're seeing right now with the use of LPO1, which is their ionizable lipid that they've worked very closely with Novartis to acquire. There's a lot of there's a lot of eyes watching on that formulation and the outcomes there in the next set of studies that are upcoming and the data that's coming out too.
Well, why don't we go to your presentation, Don? And while you're putting that up, again, just remind people to go to polev.com, enter DeBakey, and you can ask us your question if you're at your computer terminal, or if you're by phone, you can text us at 37607, text DeBakey, and ask your question, and we'll deliver that to Don. All right, Dr. Johnson, innovative lipid solutions for drug delivery systems.
Well, once again, thank you, everyone, or thank you, Don. Thank you, Christopher, for the opportunity to talk about some of the materials that we provide at Croda, but also talk about this exciting space that really is definitely something I get really overjoyed about to talk about, and that is working with LNPs. At Croda, I do want to mention we not only provide or supply excipients in the nucleic acid delivery space or the RNA therapeutic space. We also provide excipients in these other spaces, small molecule, protein delivery, and then we also sell adjuvants as well. Croda Pharma just recently acquired, in the past 5 years, a company called Avanti Polar Lipids, which was based out of Alabaster, Alabama. And this was as a result of a collaboration with Avanti Polar Lipids as a result of the SARS-CoV-2 pandemic. Sort of understanding that road, where Avanti was up until the acquisition, and sort of where things are now with the company. So Avanti was started back in the late 60s by a gentleman, Walt Shaw, who was a chemist himself. I'm trained my background is in chemistry and biochemistry, so can appreciate his interest for generating and supplying, high purity, high quality lipids. And so, that obviously over the course of the years really was well recognized by the space, in terms of us acquiring and starting collaborations, for example, with Acuitas, and then also being looked to as a resource for providing some of the key lipids that went into the mRNA COVID-19 vaccines. Since then, there's been continued interest in enhancing this, capability, where we have built out a commercial facility that everyone in my team calls it's almost like a spaceship. It's a large facility out near the Penn State area, our Lamar facility, which will go to support a lot of our commercial activity. And this is, I'm talking 50 to 100 kilogram materials. This was something that we worked very closely with the United States government in their pandemic preparedness efforts that they've put forth through a series of grants and what have you. Since then, we have not moved forward. I know on this slide here, this is a little bit of a dated slide, so I apologize. We have not moved forward with the commissioning of our Singapore applications lab. That is something that we put on hold and we're reevaluating. But we are currently reviewing ways in which we can, better support our clients and customers. So delivering across the life cycle, we're one of those few companies that can really do this. We can start very early on the milligram scale with a lot of our lipids, our lipid materials. We even provide services when it comes to not just helping some of these customers formulate their own particles with our formulation services portion of our group, but also as well with some of our custom lipid synthesis capabilities. And that's as a result of having a full in-house suite of capabilities surrounding, development of novel lipids, not to mention, lipids of interest by a lot of our clients and partners that may be a part of their own IP strategy. And so, yes, we can deliver as small as milligram quantities all the way up to hundreds of kilogram quantities, and even can be a commercial supplier of these materials for our clients. And that's really as a result of a lot of what's happened in the past 5 years. Our catalog is pretty vast. I would say we're at over 2,000 lipids, and that's as a result of having more than 15 synthetic chemists on site, 12 of them being PhDs. We have additional lipids that we typically they're part of our custom synthesis that we may have made more than we need, or lipids that may have cycled out of the catalog itself. So if you happen to look at the Avanti research catalog and you see a lipid that or you don't see a lipid that you would like, but is pretty popular in the literature and what have you, more than likely we've made that lipid. So don't hesitate to reach out and ask us, inquire, because most likely we'll have that lipid in supply. If not, it would go under a custom synthesis, which from our perspective is obviously exciting to have to go after. We have a number of different types of different phospholipids, cationic lipids, functionalized lipids, neutral lipids, different types of sterols, and obviously polymers and polymerizable lipids. Tongue twister there. We obviously work very closely with our clients and customers within the United States, but do supply through Merck when it comes to materials if it's anything outside of the United States. So that's why whenever you go to the Avanti Research site and you go to click, and there's the prompt saying, are you from inside the United States or outside? That's where some of that's coming from. So, key applications for our technologies for our lipids, right now at the top of the list for almost everybody out there, it's the LNPs. It's using these lipid nanoparticles, which I'll get deeper into. People in the past have gone with what are called lipoplexes, making cationic nanoemulsions for transfection purposes, or looking at liposomal particles as well. And so LNPs, I like to sort of throw this up here to kind of explain why LNPs, one of the reasons why LNPs have become exciting, prior to SARS-CoV-2, was really sort of from a development standpoint, I believe, outside of the therapeutic standpoint, for being able to package larger amounts of nucleic acid materials, versus using viral vector technologies for delivery purposes. The sort of immunogenicity issues that come as a result of using these payload systems, these viral vector payload systems, the ability to look at reduction in complexity, and once again, I just wanted to highlight a second time over development times. Working in pharma for as many years as I've worked in pharma, reducing development times means accelerating drugs to the clinic or to the patient at the end of the day, that are obviously safe and effective. And so these LNPs are traditionally four-component systems, with the fifth component being your active pharmaceutical ingredient, which is the nucleic acid construct. In most cases, it's a RNA construct. But the four components typically are your PEG lipid, a sterol, so cholesterol is usually used for enhancing uptake. I also believe for improving the sort of nascent structural stability of the particle as it's being formulated. Cholesterol seems to be important in that process as well. And also important in the process of formulating these particles, definitely is your cationic or ionizable lipid, in particular ionizable lipid, as it pertains to the pKa of the LNP when it's being formulated. Neutral lipids, structural lipids, these are becoming more commonly popular, especially when we start to look at what's known as tissue tropism. So tissue tropism is probably going to be one of the big important factors for what really drives this space to be, sort of next level exciting in terms of targeted delivery. And this is all really sort of driven by what we've seen out of, starting from Peter Cullis's lab when he first reported on the protein corona, and then, subsequently since then, a lot of work that's been going on between, say, the Dahlheim groups, the Michael Mitchell groups, Sehe, what have you, all of which have been sort of investigating the use of structural lipids for potentially passive delivery, for being able to dictate that protein corona. And not to mention just that, I can't, I can't help but leave out Siegert, and, his obviously his sort technology, which changes up this entire four component system in a lot of ways to be able to induce an either negative or positive charge that allows for direction to, say, lung or liver, right? So being able to get beyond the liver, as Dr. Cooke has just previously mentioned, which is kind of one of the issues right now in this space, especially when using lipid nanoparticles. And so there are a number of different ionizable lipids. You can almost throw a stone and run into an ionizable lipid in the lipid space. that's owned by, or a portion of the structure in terms of the family is owned by some company or entity in the space. There are these dendrimeric lipids, as I am pointing out here, or lipidoids, which are on the left here, or these more uncommon synthetic-based lipids from, for example, here I'm pointing out to the Sabnis group at Moderna Therapeutics, who started using the ester cleavage, acid-based cleavage technology that allowed for improved clearance of a lot of these lipids. And this sort of led to what we see now as the ALC 315s, the LC 307s, the 535s, and the LPO1s of the world, with these ester linkages on the end of their, or embedded in their aliphatic tails. And so I just kind of wanted to highlight, because obviously I'm talking we're talking at the DeBakey Center here, and Houston Methodist cardiovascular space. There have been efforts to go after cardiovascular diseases, using LNPs that are still very early, but promising. And there is some hope there to see, as a result, what comes out of companies like Verve, CRISPR, Tessera. The list goes on, but definitely exciting to see what the outcomes are from those studies, and how we can apply those to other sort of disease indications.
Don, while you have that slide up, there a couple of questions have come in, and one of our attendees is asking about ionizable lipids. Mentioned specifically alpha 031 and SM102. Now the question is, how do those ionizable lipids enhance LNP stability and delivery efficiency?
So alpha 301, I'm not, ALC 301, I'm guessing is what
ALC
you're referring to.
301 is probably what is meant, yeah.
Yeah. So SM102, ALC, ionizable lipids, all these ionizable lipids, the way that they operate is that when the particles are formulated, they're formulated at a lower pH, and so that pH on purpose is below what's called the pKa of the ionizable lipid, of the head group of the ionizable lipid. And so the charge character on the head group of that ionizable lipid causes it to complex with the nucleic acid construct as a result. And so then that helps with aggregation, precipitation of the particle, and therefore stability of the particle. So then over time, what happens is when you go to adjust that pH, go from say pH 4 to pH 7, you're slowly but surely getting the pKa, transitioning the pKa of the ionizable lipid back to a neutral state that allows it to essentially harden and properly complex into a solid LNP. One of the other factors that's happening from a solvent perspective is that you're also removing the ethanol that was introduced as a result of the lipids into the system. Typically, the ethanol isn't really causing any sort of reprecipitation or sort of crystalline effects, but it from a stability standpoint, overall, at the end of the day, you want to be able to have removed most of the ethanol.
And thanks for that. And another question that came in was about the pegylation, the PEG lipid, and the question is, how does that influence immune evasion and cellular uptake?
So PEG can cause detrimental problems for both cellular uptake and from an immunogenicity standpoint, can cause issues in the body. We've seen this so far with the vaccines and some of the studies and outcomes that happen. where too much PEG lipid actually while it can improve circulation times and obviously improve stability of the overall particle, it can be too much PEG could be a detriment for eventual delivery and uptake into the cell of interest. So there have been obviously alternatives. We could talk about some of those alternatives actually that Croda has available in our catalog. Maybe there'll be some additional questions from there too as well.
The vaccines were found to be very safe and effective, the SARS-CoV-2 vaccines. But as you point out, there were a minority of patients that had some side effects. And I guess what you're suggesting is possibly that was related in part to PEG, polyethylene glycol. But that is a very commonly used compound in drugs, in all kinds of medications. Some patients, my understanding, is develop antibodies against PEG. It's a small percentage of the population, but I guess in those patients, would you expect because they have PEG antibodies, would you expect those PEG-related lipid nanoparticles to be cleared faster? Are they going to be less effective as a result of those antibodies that some patients have against PEG?
Yes, I would agree that is the case, that you'll see faster clearance times as a result of the fact that those patients have developed antibodies against the PEG lipids. That's why we actually offer what are called these polysarcosine alternatives over PEG, and that, you're correct, is as a result of PEG being pretty prevalent as an excipient, as an additive, as a component in our formulations, our drug formulations, especially in the oral space, and what have you, for treating patients. And you're correct. I do want to make sure and iterate that the SARS-CoV-2 vaccines were definitely safe and effective. This was just something that happened in a small population, a small portion of the population.
This is the first time I've heard of the PEG alternative polysarcosine. Is that what you call it? Tell us a little bit more about that. And is it have FDA approval? Is it have safety toxicity studies in humans?
So polysarcosine is still pretty early on. We haven't really seen any first-in-human studies come about just yet that have demonstrated sort of meeting that clinical threshold for really, really getting excited about it. So a lot of the work I've seen so far has been more preclinical, pre-IND sort of level type of studies. And I've also seen a lot of the work, moving up from academia into a lot of these smaller companies too as well. So it's still very nascent in terms of, starting out. But yes, I can sort of move forward in my slides to that structure here. This is the polysarcosine structure that I was referring to, that is an alternative. It is a popular alternative, I would say, for a lot of our customers. It seems like, in what I've seen in the space, to using traditional PEG lipids.
Mm-hmm. Interesting. Okay, thanks for that.
Yeah, of course. I did want to kind of just go back really quick, point out, we were talking about the importance of the ionizable lipid in terms of stability of the particle. The process by which these LNPs are formulated is, traditionally, or in most cases, has been using a laminar flow-based mixing method, in which you have two solvent streams. You have your aqueous stream, and you have your organic stream. Your organic stream containing your lipids, your aqueous stream containing your nucleic acid construct. And so these particles they end up getting formulated at pH 4. I've done a lot of work in the past, in past lives, especially when I was at BMS, and really understanding the mechanism of action when it comes to how these particles, structurally look, how they look, and how they transition, how we're able to transition them, and what that mechanism looks like in terms of transition into the final hardened particle that we all see on the cryo TEMs whenever we send off for material. During that process, obviously ethanol is removed. This can be done using spin filtering. You can also, a lot of people use what's called tangential flow filtration, or some people like to use dialysis, which is not necessarily a very manufacturing friendly method for when you start to scale your therapeutic, but obviously still does the job in removing the ethanol while driving the pH of the system to a necessary pH that finally stabilizes these particles. And so analysis tools wise, people use techniques such as dynamic light scattering, which really gets at the size of these particles, which is very important towards cellular uptake, not to mention circulation in the body. Looking at what's termed as the nucleic acid encapsulation efficiency. Folks look at zeta potential. Some people like to look at what's called the pKa of the LNP. So that is collectively, what is the pKa on the surface as a result of these ionizable lipids, and how that sort of drives what would be the uptake and endosomal escape mechanism that takes place within the cell. Structurally, I really personally love to look at the structure of these particles. There's a lot of things that you can learn from just looking at these particles under an electron microscope. While it's expensive, while it's tedious, it is a lot of data that allows us to understand what's going on in terms of interactions between these nucleic acid constructs and our lipid excipients. In the future, if anyone's ever interested, I'd love to spend time to talk further about that. There's been a lot of work that I've been involved in that space, and really exciting work as well. Making these LNPs, there's a lot of handles, as I said before, for making these LNPs. You can undergo a technique which is known as design of experiments, which has been around for ages when it comes to developing formulations and understanding your different components and factors and what have you. And so, think of this as it could be a three-dimensional system in terms of what we call a full factorial system or a fractional factorial system. But you can also look at it in a lot of ways. What normally happens with looking at these LNP systems in terms of developing experiments or formulation panels to screen are what are called mixed DOEs. What typically happens there is you have your three factors, and you will use like one of your factors, your fourth factor, like cholesterol, for example, here as what is known as a floater, to be able to adjust your factors within the space. And then, a large, parameter to really keep in mind also as well is the N to P ratio too in the process. And so when these particles are formulated, cleaned up, ready to go, and they've been injected, they traditionally circulate in the system. There is some shedding of the PEG lipids, and that's actually was something that was developed early on when LNPs were first, coming about as a part of the mechanism to basically improve cellular uptake. And so that's why, the DMG PEG lipid, PEG 2000 lipid is probably one of the most quintessentially used lipids for lipid nanoparticles because of its aliphatic tail and how it's able to be, it basically dislodges from the lipid bilayer as it's circulating through the system. And so once, you have that circulation situation, you have the shedding taking place, you have interactions taking place between other oligonucleotides, peptides, proteins, really sort of forming what we call the protein corona, right? And so that protein corona could be a number of different proteins, ApoE being one of them, and essentially could have a lot of sort of directionality from a passive standpoint in terms of which tissue these particles go to. But when they finally get to the tissue, usually they end up getting uptaken via LDL, usually uptaken via LDLR-based, LDLR-mediated endocytosis. There are other sort of mechanisms also dependent on, the size of the particle that can sort of drive that uptake mechanism. But once uptaken, yes.
Can I stop you there for a moment? That's really interesting for our cardiovascular colleagues that may be listening in, that the LDL receptor is the receptor that mediates uptake of these lipid nanoparticles. And there are drugs that we give to our patients that influence the LDL receptor expression, such as statins. Statins are very widely used and will increase LDL receptor expression on the liver. And I wonder if, I'm sorry, I got it backwards. The LDL receptor, no, that's right. LDL receptors increased with statins. So what effect might that have on LNP uptake?
So, that would, it necessarily wouldn't have a direct effect if we were to say, obviously change the system to act, non-orthogonally, with the system. That's something we could look into. I wouldn't really be able to comment any further as to what the changes would be necessarily. And I apologize for that.
Yeah, no worries.
Yeah. So what I will say is that kind of going back here to the thought that I was going through, but yeah, that's a good point, Dr. Cooke. Thank you for bringing that up, and I'll have to get back to you on that. That is an interesting point to mention. These particles are uptaken into what's known as the endosome, and during the trafficking of the endosomally encapsulated LNP system within the cell, the pKa of the endosome is actually, or the pH of the endosome is dropping such that it drops below the pKa of the ionizable lipid. And what that does is it changes the phase of the structure itself into what is known as the H2 hexagonal phase. And the H2 hexagonal phase drives the eventual release of your nucleic acid material into the cell, which then allows for what is known as endosomal escape of your mRNA, and then obviously, goes on to deliver and function for what you're planning to either edit or drive in terms of protein expression or what have you. So there are a number of factors driving innovation in the space. We've talked about back and forth, stability. We've mentioned, the PEG dilemma. And then I've brought up some comments around targeted delivery, and, ways in which we could look at different payloads. And, also some alternatives to routes of administration that, obviously we've seen some progress in the space, especially in the annihilation space, that potentially could can be improved by some of the excipient choices that we make. And so from a stability standpoint, obviously a lot of people like to use DSPC, but from a structural lipid standpoint, there are a number of different structural lipids, and I would say, Avanti's catalog being one of the vast, sort of most diverse a number of opportunities and options there that you could look into for potentially being able to identify a number of different structural lipids. And what's important too about these structural lipids isn't just sort of how they sort of drive protein corona, but really also how they seem to drive the overall density of your bilayer, which actually sort of plays a role with understanding not just from a protein corona standpoint, absorption of proteins and other circulating biomolecules and what have you, but also sorry, I keep getting a message. I'm sorry, people keep seeing a box pop up on my right. But also being able to sort of drive stability of the particle during circulation. So we have DOPC, we have DOPE. There are a number of other PC and PE lipids that are available within our catalog. If anyone's ever interested in reviewing with me, but a lot of these lipids have been shown to have provide sort of outcomes that were not necessarily well known in the past, but have definitely been noted since we've been formulate a lot of these different companies and academic institutions have been formulating LNPs and looking at them in vivo, especially with these PE lipids and how they promote the driver of that hexagonal phase structure when it comes to endosomal escape and release of the mRNA payload. So, looking at ways we can challenge our uptake, challenge our complexing of our particles, and that being with looking at fixed cationic systems versus ionizable systems, or the combination of the two. I've seen lipid nanoparticles where they've been formulated with cationic and ionizable lipids, and that being a ratio that is looked at and worked with very closely. Certainly the advantages, obviously, of cationic lipids is that you're going to sort of increase initial uptake. This is one way of getting around that LDLR-mediated uptake mechanism, Dr. Cooke, as you mentioned earlier. And that is something that has been explored in the past. I've seen in some clinical programs. For ionizable lipids, this is probably one of the most stimuli-driven excipients in the entire system, the entire LNP system, and is very important not just for complexation, but also for eventual the end result delivery and release when it comes to endosomal escape. We've seen a lot of folks using these multivalent systems, which are these polycationic lipids more recently, such as MBL5. Folks seem to be very interested from a oral formulation standpoint of using MBL5, which is also very interesting, as the idea there is to look at ways to bypass the gap junctions in the gut intestine and increase bioavailability. So these are additional fixed cationic lipids and ionizable lipids that we provide, that we have in our catalog and are available to folks out there. We talked about these polysarcosine lipids as an alternative to PEG. This is something that's still very early on. You see a lot of folks in the preclinical pre-IND space still, flushing out the final formulations, lead formulations to carry forward into their clinical trials. I wouldn't want to comment on anything that seems to look promising just yet until it's publicly known. But this is something of interest that is a hot topic in the space. And for getting at that extrahepatic delivery, something that obviously Dr. Cooke had mentioned, we have a number of different reactive moieties that we use, these are some of the most quintessential, but not really limited to. We have DABCO, maleimide coupling, using sulfhydryl chemistry and Michael addition. DABCO, obviously going back to using click chemistry. Interestingly enough, I'm in the San Diego area right now, so a little shout out to the team who developed that click chemistry. And we provide the carboxylic acid for generation of the NHS ester. We don't normally sell the NHS ester portion of that lipid, generally because it's just not stable for shipping and what have you. It's better to just sell the carboxylic acid and the person works up that NHS ester for their own coupling interest and needs. And like I said, this is not limited, shouldn't be limited to what's here. We've seen a number of custom requests around looking at different reactive moieties, ways in which people have looked at increasing yields, looking at alternatives to improving their uptake or improving their circulation times, and that being with looking at sort of a second stimuli response driver here. Any questions?
I think, are you coming to the end of your talk then, Don? Or?
Yes.
Okay.
Yes.
I do have a question related to what you just showed us, the targeting, using these functionalized lipids to target different cells. And one of the potential indications that looks very promising is lipid nanoparticles that can target T cells so that you can deliver mRNA directly to the T cells to create a chimeric antigen receptor T cell in vivo, bypassing all the very difficult ex vivo genetic modification. That's such an expensive therapy, CAR T cells. But if you can develop LNPs that target the T cells, then you can deliver the CARs in vivo and modify the T cells in vivo therapeutically. What's going on in that space?
Yeah, so there's been a lot of really, I would say, evolution from what I would consider or term as ex vivo CAR T development to in vivo CAR T. I still think that the space is very early. There are advantages to using LNPs over what has normally been the process, viral-based process for generating that chimeric antigen receptor on the T cell. And early on, I actually was a part of a program back in about 6 years ago where we did a lot of work with using LNPs for as a reagent for being able to deliver that CAR receptor and engineer the T cells for then delivery to the patient. There are several companies. There was actually just recently an acquisition of Therapeutics that's big time in this space by Roche. So folks are familiar with Genentech. That's part of the Roche family of companies. Poseta is located actually down in San Diego, where I am right now. And outside of Poseta, for in vivo CAR T, what a lot of people have been looking at and what I've seen come up, very exciting data so far. We'll still see how it moves further along into the clinic and what have you, is from the folks at Capstan Therapeutics, who are also actually located out here in San Diego area. But their interest there is to be able to drive at what you just described, which is an in vivo based method of delivering LNPs to the patient so that their T cells are then engineered within them rather than doing it from a sort of an ex vivo perspective.
I want to turn to Dr. Brannon for a minute because you've shown us all of the various lipids that can be used to generate lipid nanoparticles, and modifying lipid nanoparticles you mentioned to us can allow for some tissue targeting. although you don't know until you actually do the experiment, right? Dr. Brannon has developed an approach for rapidly assessing lipid LNPs, lipid nanoparticles, for their tissue distribution. You want to talk about that?
Sure, yeah, I guess I'm just kind of curious what you think about sort of more unbiased approaches for screening larger libraries or larger spaces of LNP formulations. You guys are maybe at thousands. I'm curious about like into the 10,000 or 100,000 size range of LNP formulation libraries, like in an unbiased way, to get a readout of what is possible for extrahepatic delivery, and how to sort of do that in an unbiased approach. Like what's sort of the endpoint measurement? Is Croda doing these sort of unbiased screens, or is it more sort of like focused on rational development of protein coronas or conjugations or things that can, in a more rational way, direct targeted delivery, and what you might have to say about that. And I can then describe sort of a platform that we might have that would be useful for screening at the single cell level translation, of nucleic acid payloads.
So you just brought up one of the points that just gets me out of my seat excited. I'm just going to point out, and that is really, I think the way that we design our formulation screens, like you said, the approach normally is, you go after, say, 100 different combinations or formulations, but rather moving that beyond, say, those 100 different combinations or formulations and going to, say, like the 10,000, different combinations formulation scale, right? And in the process of doing that, expanding your design space in such a way that then you're able to make what are known as nonlinear sort of assumptions or speculations that allow you to immediately identify a formulation that's ideal for passive delivery and in direction towards a specific tissue of interest, especially when you're going extrahepatic for extrahepatic delivery. We just really supply a lot of these different lipids. We don't really, at Croda, we don't formulate these LNP systems. I'm speaking from the experience of what I've done in the past in terms of platforms I've developed to sort of get at this space. But I will say that the big bottleneck is being able to take those lipids, those formulations that you've made, and transition them into understanding what your readout is in vivo.
So how would you do that, Dr. Brannon? How would you do a high throughput screen to assess the tissue distribution of various lipid nanoparticles? Yeah,
we're trying to set up some collaborations with folks at Rice and here at Methodist to do those sort of screens. One of the technologies we're interested in using is single cell ribosome profiling by RNA tagging using small ribosomal subunits. So we have some in vivo systems to measure translation at the single cell level by ribosomes, and that's sort of the final output of the mRNA delivery setup. And so the idea would be to take thousands or hundreds of thousands large libraries of LNP formulations, and then just to do pooled screens with single cell readouts of translation in multiple tissues in vivo, and see if you can identify in a more unbiased way which LNP formulations correspond to effective delivery, and then finally endosomal escape and mRNA translation. So that's something that we're trying to set up a system to do, and in collaboration with some other folks, we could talk about that offline a little bit later, and how Croda might be able to help with that.
And we certainly need better delivery vehicles to the heart. We need better delivery vehicles to the vasculature. And so something like you described, a high throughput screening method would be very valuable to detect those LNPs that are preferential to different organs.
Right.
Yeah, I would agree. Looking forward to those discussions, Dr. Brannon.
Some other questions came up during your talk, and one that came up was about the functionalized lipids, your ability to target antibodies to a lipid nanoparticle and thereby target a tissue. I guess that's another way that you could rationally design a lipid nanoparticle to target the heart, for example. By tell us about the lipids that you have that could allow us to do that.
So we, like I mentioned before, we have the DAPCO-based approach or bulamide for coupling purposes, but we also have a number of different options, alternatives for our PEG lipids. So, for example, we have DMG, DSPE, ceramide-based type structures, classes of structures that could be investigated further with our clients, in which those structures, have different sort of near neighbor interactions, obviously, and more, structural interactions with other components that allow for stability and therefore proper circulation in delivery times. So we have a lot of those lipids that are available, and we can obviously take, that person's very interested in, digging deeper, looking further into those structures. We can work with them offline.
We have maybe time for one more question, if you don't mind. You mentioned at the very top of the show that you're a cancer survivor, and that drove you into this field. And I think one of the things you must be finding of great interest is the development of personalized neoantigen therapy, cancer vaccines. And we have a program here at Houston Methodist that we're developing personalized cancer vaccines for patients with triple-negative breast cancer, but there's all sorts of possibilities for that therapeutic. Tell us what lipids might be best for that kind of purpose, for personalized cancer therapies, neoantigen therapies. You have any thoughts about that?
Right now, I would say, some of the work that I've done in the past when it came to, the CAR T cell development aspect, those are using more conventional systems. So like the D-Len KC2, D-Len MC3s of the day, those more conventional lipids may be a starting point. I would argue for if we were to go into direct RNA therapeutic space, combinations of structural lipids, not just say one structural lipid. I don't think you can just formulate an LNP with DSPC and keep moving, but rather if you complicate that system with say a 5-base or a 6-base system beyond a 4-base system, you're going to be looking at a lot more fruitful combinations of therapeutics, of formulations that are going to be useful as a therapeutic.
Well, thanks for that, Don, and thank you so much for sharing your wisdom with us on lipid nanoparticles, very critical for what we're trying to do with RNA therapeutics. So thank you for joining us on the show. I want to thank my co-host, Dr. Christopher Brannon here at Houston Methodist and our Center for RNA Therapeutics. Thank you all for joining us to hear Don Johnson talk about the latest in lipids and lipid nanoparticles. Thanks again, Dr. Johnson, for joining us on the show.
Thank you.